Okay, so hi everyone. This is the fourth condensed lecture for protein algorithms to describe protein structures, and we're going to talk about scoring functions today. So in the previous lecture we talked about the different strategies to sample conformational space to predict protein structures based on, for example, homology, or in the case based on threading, or in the case we know nothing based up ab initio methods. And now we're going to see how we can evaluate the conformations that are generated through these strategies. So the first class of methods, let's say, is the first class of scoring function are those that rely on physics, a physical description of the biomolecules. So typically these scoring functions, these functionals are called force fields. So you have here an example of such a force field. So these were developed in the 80s, let's say. The development of these force fields started in the 80s, and you have different flavors of these different force fields. So for example, this is the amber force field, and generally you have one force field which is associated to a package allowing to perform molecular dynamic simulations. So the parameters between the different force fields are going to be different, but the main shape, the main elements remain the same. So what you see in this formula here is that you have terms that correspond to interactions between bonded atoms, meaning atoms that are linked by covalent bonds, that are close to each other, and non-bonded terms, which are going to correspond to interactions between atoms that can be very far away, even solvent atoms. So the three first terms correspond respectively to the length of the covalent bond between two atoms, the angle between three atoms that are linked by two covalent bonds, and the the dihedral angle, the torsion angle between four atoms that are consecutive, that are linked by covalent bonds. Okay, so the two first term are have a harmonic form, so it's just a harmonic potential where we approximate this bond or this angled with a spring, essentially. So you have K, which corresponds to the spring constant. And so this is a sum over all the pairs of atoms that are linked by a covalent bond, all the triplets of atoms that are linked by covalent bonds, and where you can define an angle, all the ensembled sets of four atoms that for which you can define a torsion angle. And then the non-bonded parts. So here the sum is over a much bigger ensemble, because you will have interactions between atoms that are not related by any covalent interactions. So essentially they can be anywhere in the protein. So we're going to give a bit more details into those terms. But right away you can see that the complexity of the calculating these different terms is not the same. So for the bonded terms, the complexity will be linear. Whereas for the non-bonded terms, you have a quadratic complexity in principle, and methods have been developed to achieve logarithmic complexity, n log n complexity. So an important part here is that the parameters which are shown will depend, in fact, on the types of atom you are dealing with. So every type of atom will be associated to a different parameter. So that's a lot of parameters. And these types of atoms are not the elements, the chemical elements, but they're really, it's more precise than just chemical elements. It's really the valence, the position, the interactions they make, etc. So these correspond roughly to the atom names that you have in the PDB file. Okay. So if we go a bit more into details, first, the covalent bonds. So the potential energy that is associated to this bond in principle. So here there should be an animation, but okay, you cannot see it. It's just an example of water molecule and the type of motions that you can have with respect to this covalent bond, how it can move. So there is the symmetric stretch or asymmetric. So one does like this, and you can have some bending of the molecules. So in principle, the potential that should that correspond to this bond is represented by the green curve that you see here, which we will talk about this curve a bit later on. But for practical reasons, so that's Moore's potential. But for practical reason, and especially because this Moore's potential, you need to take energy values that are discrete. We will simply approximate it with a harmonic potential. So that's what I said before. You just associate or just represent this covalent bond by a spring with a constant. So you basically have a quadratic form for the potential. That's just too well what you see in green. So these are the different terms and how the potentials look like. The first one are quadratic terms, and then you have the dihedral, and here you have a sinusoidal function. Okay, so this, I will go through it quickly. So then for the non-bounded parts, so when you've done this, you essentially have all the local interactions. Then for the non-bounded parts, there are really two main forces that we consider, types of interaction that we consider. The first one is electrostatics. So simply if you have a plus and a minus, they will attract each other. If you have two pluses or two minuses, they will repulse each other, right? So this is represented by a Coulombic potential in the force field, in the functional. So you simply have a multiplication of the charges that are in each atom, in each particle, and you divide by the distance between the two atoms and also some coefficients which represent the permittivity of the void, for example. And so what you can see here is that the energy will decrease when the distance between the two atoms increase, but it will decrease slowly, let's say. Now the second term is reflect the van der Waals interactions, which are non-electrostatic interactions that will occur between dipoles. So if you have the spheres here that are shown, right, represent the volume that really each atom takes. So you can represent an atom by just a coordinate, 3 coordinates, just the center of this atom, but in fact you have a volume that you can define around, which represents the volume taken by the atom. So this is defined by radius, by typical radius, which depends again on the type of atom you have. Okay, so if you have these two balls that are shown here, they really bump into each other. So you have a steric clash. So this would be really unfavorable from an energetic point of view. Okay, so if you're too close, you would like them to be repulsed from each other. So in the potential that we're going to consider, we will have a term that represents this repulsion. And then if you have two atoms that are, let's say, close to each other, but not too close, they will try to attract each other, right? That's the weak dipole attraction between distant atoms. So this is an attraction term that you will have also in the potential. So basically you have some parameters on top, and they're divided by by again the distance between the atoms, but you have a coefficient. So here it's 12 and 6. These coefficients can vary, and they were defined empirically, so it's not really something you can be, let's say, sure about. But the important point here is that these coefficients are big compared to just the distance that you have for electrostatics. So meaning that this force compared to the electrostatic is short range. Electrostatic will be long range, so even if you have distant atoms, their charge are going to see each other. If you have So they will tend to attract each other and the charge are complementary, but they will not see each other from the point of view of this force. So this is just a force that will really apply on things that are close by. So if we decompose the potential, we have on top the repulsive curve. Why repulsive? Well, this can be seen easily because for small distances it's very high, so very unfavorable. And the attractive part is very favorable when you're close, and then it will just reduce to nothing. So you have essentially an equilibrium distance where you are the most favorable possible, right? Meaning that you are not too close but not too distant. So you don't have you're at the minimum of the repulsive and also the minimum of the attractive. So the attractive gives you the favorable part, and the repulsive is just neutral. Well, it's not exactly the minimum, but let's say it's acceptable for the repulsive part. So if you combine two curves, then you end up with a red one, right? And okay. And so again, as I said, there are a lot of parameters, let's say, that you need to fix. So how do you define those A, those B's, etc.? So for this, what you can do is to use experimental data coming from spectroscopy, from small molecular crystals, or to infer them from quantum mechanics calculations. So as I said before, we placed ourselves here with this force field in a context where we model or simulate the motions of proteins according to the classical mechanics framework. So the sum of forces is equal to mass by acceleration according to Newton's law, but this is not accurate because proteins are small, so we should be in a regime where we consider quantum chemistry and the equations of Schrödinger. So from these quantum mechanics calculations, which are much more precise, but which cannot be applied to very, very large systems, you can infer the parameters that you're going to use in the force field. So they are computed for certain types of molecules, meaning that you will have different parameters for proteins, different from DNA, RNA, and also compounds, chemical compounds. So they may not be transferable from one type of molecule to another. So if you develop a force field, your aim would be to be as accurate as possible, but also to limit the number of parameters you have to use and infer, and to try to be as general as possible, so that if you have a new compound or a new thing new ingredient you need to consider in your system, you don't have to redo all over again this determination of parameters. Okay, so then you can consider the pair versus multi-body potential. So Coulombic and van der Waals potentials, van der Waals, by the way, is called Lennard-Jones generally potential. I did not mention this. So these are summed over pairs of atoms. Okay, so we just consider only the pairwise interactions, pairwise couplings that we have between atoms. But in fact, we could try to model explicitly the interactions between triplets of atoms, because we can have effects like this where indirectly some guys are going to talk to each other, but in combinations there are more than two. The problem is if you do this explicitly, you have a combinatorial explosion. So there is a way to account for the influence of the other particles in the system using effective potentials. So this is to account for the screening effect that you will have because of the environment. So you don't have the true interaction energy, but the parameters allow you to include these effects of the other atoms in a mean way, let's say average way. Okay, so that's really the first class of potentials, these force fields, which are quite accurate, and but they have many parameters and they have some limitations. So another route you can go is to try to rely on statistics. So for this, you will define potentials that are based on the occurrences of atom type that you see in PDB structures, and the typical distances, typical angles you see between different types of atoms. Okay. So the idea is that the native structures that we have in the PDB have distinctive geometry features and they're reliable enough to represent what we want. So we're going to infer these parameters from them. So also an assumption made here is that binding energy can be approximated as a linear superposition of individual interactions. And there is a big assumption is that the Protean distribution functions obey Boltzmann statistics. So there is a general form, let's say, where the high energy conformation, which is unfavorable, will be much less probable than the low energy conformations. So the problem with this is that the reference distribution function GR is rarely known. Okay, so this is an example of an illustration of the Boltzmann distribution. So you have different types of distributions that are shown on the plot, and the Boltzmann is the first one. So you really have this this relationship between the energy and the probability of occurrence. Let's say the red squares correspond to probability of occurrence. And you can see that as the energy increases, your probability of occurrence reduces. So it's gradual, but it's stronger. I mean, even for small delta of energy, you will have a strong reduction in probability of occurrence. So the thing that you need to keep in mind is that Protean adopts an ensemble of conformations and solutions. So there is an equilibrium between all these conformations and some probability of occurrence that we can define for the conformations. So the expression of the Boltzmann distribution is given here. The probability of a conformation I will be the Boltzmann coefficient over the partition function. So the partition function is nothing magic, it's just the sum of the Boltzmann coefficients for all the conformations. So generally we don't know it. We don't have access to all the conformations, otherwise the problem would be solved. So, okay, that's an issue, but what you can see here also is that you have a coefficient which is exponential with respect to the energy, and that's why your probability is changing a lot, while the energy is not changing that much. So popular potential that can be mentioned, the Dot potential. So these are statistical potentials. So typically the Dot potential is statistical. It's based on the distances that you observe in PDB structures between certain types of atoms, and you can consider all the combinations of types of atoms. We can mention the Rosetta potential, which is not purely statistical, but it includes a large number of terms, and you have some of them physical and others statistical. So let's say it's an empirical force field, an empirical scoring function, energy function. And okay, I don't talk too much about it. You can check the paper. So then you can try to go a bit further, and nowadays it's maybe becoming more and more relevant as we have more and more data. Instead of doing simple statistics, let's say you can try to learn based on the data that you have in maybe more sophisticated ways. So the idea is to get rid of any a priori that you may have on the system, and just try to have some very generic functions that are going to be tuned on the data. So this would be an optimization problem, which can be compared to the Boltzmann inversion. So you don't have anymore this. You don't need this assumption according to which the conformations are distribution according to Boltzmann distribution. So there are pros and cons for both. Okay, so for the optimization, the reference state distributions are not required. There is no assumption about the weights of the different contributions, and you can use potentials that are smooth differentiable by construction. But you need to be careful on the way you design your training sets. Okay, so you need to generate false examples. You cannot just learn from positives just from the PDB, because these are not just statistics. You need to be able to distinguish what is a protein from what is not a protein. And you may learn peculiarities, you may overfit. The Boltzmann inversion is well suited for protein folds, where you have reference state that can be computed. And you can do many body statistics, but there are other problems associated to this. Okay, so you start from an ensemble of structures and you're just gonna fit your curves, let's say. So here is an example of potential that is derived from learning. So if you can say data driven. So the idea here is that you compute a number of properties on your tertiary structure examples. So for example, residue pairwise density functions, atom pairwise density function, hydrogen bonds. So you have descriptors. So it's not exactly true to say that you don't have a priori, because you have some descriptors that are linked to the chemistry, to the object you're looking at, of course. And so you integrate all these parameters, for example here, so you have normalization step or scaling step, and then you use, for example, ridge regression to end up with a score, which will give you the quality, which will indicate you the quality of the model you're looking at. So the local quality, or and then you aggregate these local scores to, for example, to the whole protein. So for this, here, for example, in that example, you have the dimensionality of the feature space, which is huge, 4 million parameters, but very sparse. And this sparsity allows you to learn efficiently. So then you can also use convolutional neural networks. So what will you do in that case? Well, this is a very generic slide about convolutional neural networks, with the classical example for images. So you have your image and you have some characteristics, and you're just gonna make some convolution on this description, this 2D matrix description of your data. Different operations, so convolution, subsampling mainly, and also applying some nonlinearity to provide the output. So for example, you can do this in 2D. This has been applied also on sequences. So here the objects that you're looking at may be conformations, but they may also be sequences. So you can use protein sequences to infer protein structures. So here you have an example in 2D where the input will be multiple sequence alignments, and you will derive contacts between residues, contacts between positions, or even distance between positions. So this has been done in the past years, and this has been actually a major revolution in the field. So the first revolution was really to realize that you could leverage the crazy amount of sequence data that we have now to predict some contacts, to predict 3D structures. And the second revolution was to to boost the performance using convolutional neural networks. Or you can still you can work with objects that are in 3D. So where you're working with the structures. So this is an example of here you have an example where you are gonna represent your system with different maps, 3D maps, which correspond to the density of atoms you have, different types of atoms, and you learn on these maps. And you may also think of graph convolutional network for this purpose. So I'm just gonna go a bit more into the effect of coevolution. So the fact that what we can learn, what we can get from sequence data. So for example, here you have some sequences, they're on top of each other, so you have a multiple sequence alignment basically in a different species. And I just highlight two positions. What you can see in these two positions is that there is a switch of the amino acids at these positions, and the switches are correlated. So I have a K which becomes an E, and an E that becomes a K. So meaning that if you have an interaction between these two residues which should attract each other because one is positively charged and the other is negatively charged, then there will still be a possibility to maintain this interaction even though they're mutated, because the mutation is compensatory. So you may think that you can exploit sequence data by trying to find these co-evolution or co-variation signals between different positions, and that they will affect and that they reflect some functional dependencies between positions, and even more some structural proximity, 3D proximity in space. So there's been a method developed by Martin Weicht in 2009 to be able to infer these contacts, these signals with high precision, and to disentangle some direct covariation from indirect covariation. And it's been shown that these signals correlate very well with 3D proximity, so that the physical context that you have between the residues in the 3D native structure. So here is an example of a science paper which was published in 2017, where protein structures were generated based on contacts predicted using coevolution signals found in genomic and metagenomic sequences. So you may use genomic sequences, but you may also use metagenomics, which are chunks of sequences, small chunks of sequences. So if we look at the evolution of the performance in structural prediction, so for example here we have a plot showing different instances of a competition, which is the CASP competition, which is an international challenge to determine protein 3D structures. You can see that the performance increases over the years here, right? And you can really mark the emergence of coevolution-based predictions of contacts. So this is around CASP 11. So at first these predictions were made in an unsupervised way, and they were improved by going from unsupervised to supervised. So basically you learn over some maps that, so some contact maps that are, so you learn based on your inputs is still the multiple sequence alignments, and your output target is are the contact maps that you know from the PDB structures. Then you have the big boom thanks to the deep learning architectures, and we can mention RaptorX method, which was really very successful in this. And more recently, so this is from last year, I guess, was it last year or the year before? The DeepMind team came on the problem, and they developed AlphaFold, which essentially just takes the ingredients that were already there, so coevolution, leveraging sequence data, and deep learning. But they used it in a way that was maybe a bit more sophisticated. Well, they predicted not only contact but distances, so they went from contact to distances, and they also had some maybe other features or other way to deal with auxiliary outputs, auxiliary networks, with also a way to chunk sequences in a very systematic way. So in any case, they were able to really increase very much the performance. Okay, so just to finish, I would like to, on this part, I would like just to de-zoom a bit and ask yourselves how close maybe are we to numerically solve the protein folding problem. So just to give a broader perspective on the history of the problem and on what it means to solve this problem. So two things that can be mentioned. The first one is the production of single molecular dynamic simulations of the folding of 12 small proteins at realistic timescale. So this was done by the Show Group in 2011 with a supercomputer which was designed specifically to run this type of simulations. And so what they saw is that native-like contacts are formed in unfolded state, and the successive stabilization of key contacts are driving the folding reaction. So this was a big step at the time because it really allowed to see these proteins in 12 is not nothing fold inside the box of water molecules that are explicit. So there is also a framework developed by Vijay Pandey, which is short-end simulations combined with a Markov state modeling framework. So the folding at home game, not game, it was not a game, the game is folded. So the folding at home project, where you have a community of people who just give time of their computers, and these small simulations run, and then you just let's say put them in computer transitions between them in a Markov model framework. So here you get access to accurate estimates of the kinetics of protein folding, and what you see is that they behave a bit like they have a glass-like behavior. So now, what if we compare these advances and these things that have been achieved? So, okay, these were by simulations, and I didn't mention, but of course I talked about it just before, these advances in protein folding thanks to sequence data. That's a bit different, right? Because here you really start from you're trying to fold in solution, whereas in the sequence, then you're just trying to find the correct structure without this idea of simulating in a physically realistic environment, let's say. So now we can compare these efforts with what we expect in the tube, right? So in Celico, we start from an unfolded full-length ensemble, typically, in these simulations. And the time spent in the unfolded state is determined by the thermodynamic stability at equilibrium. We have a high dilution, so we don't have any really competition or crowding effect most of the time. And we're just looking at the protein moving until it folds. So there is a spontaneous folding, no competition, no crowding, and a homogeneous environment. Whereas in the tube, so maybe in real life, well, not in tube actually, in vivo, what you expect is that you start from a nascent chain, right? So you have the process of translating the proteins by the ribosome. So there is this notion, you never start with a protein that completely unfolded and just folds. This doesn't make sense. You just start with a chain that will increase, and as it increases, well, you don't know really what's happening. Is it staying unfolded, or is it starting to fold already? So we know that small proteins will likely fold on their own, but big ones are going to be held by chaperone. So there are other mechanisms that play a role, which are not in simulations. The concentration of the proteins is very big in the cell. It's a very crowded environment. You have a competition between this folding process and aggregation, and when you have misfolding, you will have aggregation, which are associated to diseases. And we know that because of the compartments we have in the cell, there is a spatial organization, so it's a highly inhomogeneous environment. So these are, let's say, problems that can be solved by the simulations. So when you look at sequence data, it's true that you don't care about all this. You're just looking at what was produced by evolution over the years, over a very large number of years. And so, in principle, the signals that you can reveal in the sequence data account for the environment, account for all these aspects in an indirect way. Okay, so the complex environment of a cell makes the protein folding problem a completely different story, you could say, when we take it from simulation or from the in vivo. So there are many, many aspects that we have. So then we may ask, is the protein folding problem correctly specified? So how is the information for protein folding landscape encoded in a given sequence? That's really the problem. That's the question we're trying to ask. We assume that we have this sequence that encodes information to fold the protein to determine the 3D structure, and that we are able to, there is a way, there is a code, there is a way to find this 3D structure. So what about the many selective pressures for maintaining and adapting a protein function? What about the stochastic events that have led to that particular realization, that particular solution? So what we see as a result of evolution is a particular realization among many others that are possible in principle. And this path is stochastic. And what about the impact of the environment? For example, interactions with other proteins like chaperone. So all this should be maybe taken into account. How do we do? So a protein folding landscape description should consider its evolutionary history. So there is something that is really brought by considering the evolution, which even if it's indirect, it's better than nothing probably. So I will just finish with a citation. The protein sequence we examine today is not the embodiment of some ideal type or norm, but rather unique and repeatable realization in the field of quasi-infinite possible genetic combinations. So of course we know that not all the space of all the combinations can be explored because of physical constraints. But it's also good to remember that we are looking at particular and somehow randomization, realization among a set of possible. This is by Francisco Ayala. Okay, so with this I finish.